Okay, thank you everyone for joining us for this special campaign webinar for Trees Around the Globe student research campaign. We are excited to talk about and hear about, I should say, flowering tree phenology today from the ground in space with our guest speaker, Dr. Yoseline Angel. And we're super excited because we love the special webinars because they bring together things that, we've kind of been focusing on for the entire campaign, but really fine tuning, what we've been doing with the campaign. So today is scheduled for, an hour at the max because I'm not going to be doing any campaign metrics. We're going to save that for our regularly scheduled campaign webinar, which will happen later in this month on the 29th of April. But I'm going to jump right into it so we can have time for the speaker and some question and answers afterwards. So please feel free to add anything to the chat you'd like, any questions during this introduction and the featured presentation. So I wanted to start off by this little quote that I found that I think is interesting by Mark Worms, the chief executive of the Los Angeles State and County Arboretum. In full glory, the Tabebuia trees are absolutely heart-stopping. The pink are so gaudy, they're wonderful, and the golden so refined in comparison. I just thought that was an interesting quote, and this was apparently a quote when he became the chief executive of the Los Angeles State and County Arboretum, and he first discovered these trees. And I just think that's a really fun quote to get us started today. So with the agenda, we have the featured presentation, and it's entitled Revealing Hints About Dynamics in Flowering Tree Phenology from the Ground in Space, including a request for GLOBE program participants. And it was with Dr. Yoseline Angel, remote sensing geoscientist and engineer at the NASA Goddard Space Flight Center and the University of Maryland College Park in the United States. And following that, we will talk a little bit about the request. I know she's going to mention it during her presentation, but there's going to be a request for the GLOBE community for looking for and observing the Tabebuia trees and the GLOBE Observer Land Cover and Trees tools. And there are a couple phases that I believe she will mention that is part of this. And then we will finish up with a question and answer session for our featured speaker. All right, so we're going to pass it over to our featured speaker. And once again, Dr. Yoseline Angel. And she's going to be talking about flowering tree phenology and how she looks at these things from space, how we can take observations on the ground and do some comparisons. As you know, we've done with the campaign a lot with different things, from everything from ICESat-2 and JEDI to Landsat to all of these different missions that we focus on when we talk about tree height, land cover, greenings, green up, green down, carbon cycle, all the things that we focus on with the Trees Around the Globe student research campaign. So without further ado, I'm going to pass it over to our featured speaker, Dr. Yoseline Angel, and I will stop sharing. And welcome, and thank you so much for being here.
Hey, Ryan, thank you so much for having me and for the invitation to this special webinar. To me, it's pleasure just sharing our science, and not only our science, but our passion for flowers and flowering phenology, and how we're using remote sensing and developing flowering spectroscopy for the purpose of identifying, mapping, and monitor flowering phenology around the world. So I'm going to start sharing my screen here. Please let me know if you can see the presentation so I can start.
Yes, it looks great.
Okay, perfect. Thank you. So, well, thank you for the introduction as well. I'm just going to start by showing you something. This is an image that one of our sensors that is now on the International Space Station is called EMIT. It's an imaging spectrometer or a hyperspectral sensor that collects hundreds of spectral bands from the visible spectral range to the shortwave infrared. And this image that you see here in the background was collected in California last year during the spring season. those yellow areas that you see here that I'm indicating here with my mouse, with the pointer of the mouse, correspond to some flowering events that were happening last year during the spring season. So this is how major flowering events are like displayed on these images, and how we can see them from space. But let's go back to the basic. I want to share something with you. So the purpose of flowering is reproduction. So it's a mechanism that flowering plants use for reproducing through pollination. So what they do is like making themselves appealing to pollinators with different mechanisms. And one of those mechanisms is through colors that we are not even able to see because our sight is limited. So here, I can show you our human vision is limited to 3 spectral bands that cover the blue, the green, and the red spectral range from the visible electromagnetic spectrum. But for instance, honeybees or some other pollinators, although they have the same 3 spectral bands, they have one of those bands located or like targeting the UV spectral range. So what they see is totally different to what we are able to see. Even further, there are some other species, well, not pollinators, but some other species that, like the mantis shrimp, that have that has way more channels, way more spectral or bands that cover the same spectral range. So we can say somehow that they have like a hyperspectral vision or a superspectral vision. So let's just think for a second how they see the world and how they see everything around them. Well, although we are limited, our vision is limited to these 3 spectral bands or these spectral ranges, we develop some sensors that we call spectrometers, imaging spectrometers. So here I'm going to share with you the 3 different scales that we explored with these sensors, with these imaging spectrometers or spectrometers, from the ground, airborne platforms, and space platforms. From the ground, we use something that is called just a pointer spectrometer. We point down with the sensor to the canopy to collect the amount of photons that come from the sun, reach to the surface, like leaves or flowers, and these are reflected then back to the sensor. So the amount of photons or energy along the different spectral wavelengths that these sensors are able to see, we collect the number of photons somehow, or the reflectance, that give us an indication of different traits or characteristics of these flowers or these leaves on the ground. Some of them related with pigments, for instance, chlorophyll or some other pigments, or water content or some other chemical compounds within the leaves or the flowers. From an airborne platform, we are also able to see similar spectral characteristics like these absorptions or minimums that you see here along the spectral profile, or some maximums, or some other characteristics like this slope here, or just like an edge here. And these are the spectral features that we want to describe and that help us to distinguish between flowers and leaves and soil and some other elements within a pixel. From space, the principle is exactly the same, just that our spatial resolution is way less. Well, I just want to share with you something a little bit more deep, which is how the spectral profile across different colors for a species that we test in our lab look like. So you see how, depending on the color, the predominant color of the flower, we have a different spectral response along the visible spectral range, from 350 nanometers to around 1,000 nanometers. And the characteristics, the maximums, the minimums, or absorptions, the edges, depend on the pigments like betalains, carotenoids, flavonoids, anthocyanins that are present within these flowers. And these spectral features are the ones that we want to identify, we want to characterize to disentangle the spectral contribution of these within the pixel. So I'm just going to show you an example. This was our first work on imaging spectroscopy using an airborne platform in California. It was 3 years ago during the spring season. We collected a high time frequency series with 5-meter resolution over 2 different reserves where we found this flowering area with 2 predominant species that you see here, the Coreopsis gigantea and the California sagebrush. This wasn't the first purpose of our campaign. It just happened that we found these flowering displays there, and I got interested to do flowers, basically. So we collected the spectral features or the spectral signatures from the 3 different yellow flowering species, which were the more predominant at that time. And you see here a comparison of the 3 of them versus the leaf spectra of these. And you can start seeing the difference, especially here in the visible spectral range. 2 different edges, the yellow edge, and then we have this edge here, which is called the red edge. But if we, so this was collected clipping or directly making contact with the flowers, with the petal, and making contact with the leaves. But once we move and collect this from making non-contact from the top of canopy, you start to see differences. And this is when environmental factors start to play a role within our spectral profiles. So imagine how these spectral profiles will look like from even higher sensors, like an airborne base or a satellite base. This is how the soil looks like. And this is how it look like if we pull out of a pixel from this flowering area, where we have the mix of different elements within the pixel. Mostly, and just to simplify, what we will find within the pixel, it will be the mix of yellow flowers, green leaves, soil, and shadows. So we have 4 elements. We can find way more elements within the pixel, but these are the 4, let's say, most general that we will find along this area. And here, just a comparison of what I mentioned before, the yellow edge versus the red edge. The yellow edge is a spectral feature characteristic from the carotenoids, which are the yellow pigments, and the chlorophylls, which are the green pigments. So we can start targeting this spectral region to try to disentangle both spectral contributions within the pixel. So that's how we frame our pixel, let's say, considering 4 elements: leaves, flowers, soil, and dark signal. Dark signal comes from shadows or water pixels. So what we do is applying a spectral unmixing method that aims for identifying the weighted contribution of each of the pure spectral profiles of each of these elements within the pixel. So we get the percentage, let's say, of each of these elements within the pixel, plus some residual signal. And what we use for performing the classification and identification of flowers is the residual signal. Some people consider this residual signal as noise, but for us, the residual represents a rich source of information where we can find these differences between, for instance, between flowers and green leaves. So instead of using the end members contribution or the percentages, we keep going in our method with the residual reflectance, which is also a data cube with the same number of bands as the reflectance data cube. So using the residual data cube, we reduce this through principal components to simplify again the estimation or the classification. And we keep the first three principal components that comprise around 95 or more than 95% of the information, the spectral information. And what you see here, it's like just a representation on the feature space of these pixels. Each point here in this cloud of points represent a pixel. And we perform some sort of like clustering method, which is Gaussian, which means it's a probabilistic method. To estimate the probability of occurrence of certain cluster and non-cluster, because this is an unsupervised method, so an uncertain cluster within the data set. So we start like considering that we have only one cluster and it's an iterative method. So then we add a second cluster and then a third one, a fourth one, and so on and so forth, until we reach to a certain point when there's no longer need to add more clusters. And we identify that one of these spectral clusters, the one that I have here like with yellow color, corresponds to our flowering areas. So here you see the results of this flowering area that I showed at the very beginning. And you see here how dynamically the flowering area is changing over the study area. And because it's a probabilistic method, we also retrieve the uncertainty of our of this probability of occurrence, which is also great because we can assess our results. So from high to low uncertainty, or 0 to 100%, or 0 to 1. Well, this is just the science behind. This is what we are using for detecting different flowers. And maybe you are thinking already, like, why only yellow flowers or why yellow flowers? Well, this is because it happened that most of the predominant species in that study area were yellow flowers. But we can extend these to some other colors, as I show in our example, across different colors from that we extracted in the lab. Second result, and something really exciting for targeting and for monitoring phenology, is the use of vegetation indices. So using the residual signal, we develop flowering vegetation index that we call, the mixture residual blooming index. The mixture residual blooming index in this case is targeting the yellow spectral region, the visible spectral region that overlaps the yellow colors. And what you see here is 2 time series. The yellow time series is showing the mixture residual blooming index along the spring season. So we have weekly imagery from the beginning of March to the end of May, more or less, there in California. And you see, like, there are like 2 different peaks, like one that lasts short, like around a month. And then we have a second peak that lasts longer. In contrast, we also wanted to see this, we wanted to evaluate the greenness or a greenness vegetation index, like the red edge NDVI, which is a hyperspectral NDVI that show us totally different dynamics of phenology. How at the beginning of the season, we have higher greenness, and then the greenness decreases through the course of the season. And somehow is what we see here in the picture. So on the top picture, we have the beginning of the season, where we have way more greenness areas or green leaves. And then by the end of the season, we have way less green fraction there within the pixel. But in contrast, we have, for flowers, we have at the beginning high yellow coming from the Coreopsis gigantea. And then by the end of the season, the yellow contribution comes from the Artemisia californica. So this is a way to use high temporal resolution with high spectral resolution to see or to study the phenology even between two different species. Well, this was our first work using imaging spectroscopy and flowering phenology. But then we have to keep moving, like validating our results, expanding these to different scales and transferring these to satellite-based sensors like EMIT. So for that purpose, we've been using 3 different sources. One of them, commercial satellites that have high spatial resolution, 3 meters of resolution, like the PlanetScope data set, or Maxar, like Worldview, GeoEye, that some of them reach like even sub-meter spatial resolution. So we can use them, like the historical data set, we can use them to validate our results. In addition, we also use some citizen science or data sets, open databases like the iNaturalist, or in California particularly, there is one that is called CalFlora, where people or scientists register the historically flowering events. And simultaneously, and this was something that I was like thinking about, like flowers is something that most people like, that most people love. When they see that it's flowering, they immediately jump to the field and want to take pictures and selfies and share all these with the public in general. So I started to follow several trends, different trends, as you can see here, the tags, the hashtags, flowering, wildflower, super bloom, in Spanish and English both. And whenever some of these events happen, people start like publicating these or posting these on their social network, and I start to get alerts of when and where this is happening. So this is another source to get an alert of different flowering events around the world. And I've seen, I mean, now my social networks are just full of alerts with flowers here and there and everywhere, which is beautiful. I like it. So this to the moment. But then I learned about GLOVE, which is this tool that we have in NASA for doing the same, but collecting the information directly to our and saving these directly in our data repositories. So we started this year to set up one of the first data requests through the land cover form, particularly for some areas here in the United States, most of them located in California. And the reason of California is because of our different data sets that we have there, and because of some other projects that we are like developing there. But the idea is to expand these all over around the world. This is just the first test of GLOVE for the purpose of monitoring or collecting data observations from flowering events. And then using these observations for the purpose of validating our maps and validating the way we are retrieving these flowering events from satellite platforms. So I'm just gonna expand a little bit more with this project. So we call this project the Wildflower Blooms. What we request here basically is three things mostly, like a picture of the general landscape, then a couple of pictures more of specifically like a close-up from the different flowers that the people will see in the field. So we will be able then to identify the species, and perhaps in the observations or the notes, some side notes, people can also like help us like indicating what was the most predominant color of the different flowers that they see in this location, and maybe some other notes that they want to add. Or if they get to know the common name or the scientific name, it will help a lot. Well, this is part of what we are doing for shrublands, grasslands, but what about flowering trees? So I'm just going to jump into something that has been like, I've been thinking a lot recently. It's about the cherry blossoms dynamics in Japan. This in Japanese is called sakura. Cherry blossoms translate to sakura. So here you see an example of the sakura happening in the Mount Yoshino in Nara Prefecture in Japan last year. So here you see an example of the sentinel image, a comparison between two different years. To the left, we see last year. To the right, we see 2021. Then we have a planet scope where we can see way more, with more definition, even the colors of these cherry blossoms. And from iNaturalist, we learned that the cherry blossoms we are able to see the cherry blossoms all over the northern latitudes in States, Europe, and also, well, around Japan and China. And they have only one, let's say one flowering peak happening between March and by the end of May. However, this peak of the blossom has been changing all over from 1,200 years ago, which is incredible because the Japanese people have been recording the historical data from this cherry blossom peak. And what we can see here in the figure is how this peak has been changing and drastically changing during the last 50 years. So every time is like happening early than before. And somehow this is what we see here in the Sentinel image when I compared 2024 with 2021. The image from 2024 to the left is from March 15, and the image from 2021 is from March 26, like 10 days different, more or less. So inspired on this, on using these sort of flowering trees, species, to monitor climate dynamics and environmental dynamics, temperature dynamics, I thought about like, okay, we are covering with this somehow higher latitudes, but what about mid-latitudes? So this year I have the opportunity to join a campaign in Panama, the Abuelo campaign, the Airborne Validation Unified Experiment, Land to Ocean, that happened in Panama and Costa Rica, but I was based on Panama in the Smithsonian Tropical Research Institute. There we have two different purposes. From one side, like collecting, keep on collecting different flowering species to get more variation in our spectral library from different flowers, different pigments across different colors. Again, and this is an open question, and maybe maybe some botanists, plant scientists, they know the answer to this, but it caught my attention that yellow flowers or yellow pigments are the most predominant even there. Well, but we get some other colors like pink, purple, white, and some other colors there. This was one of the objectives of doing this flowering spectroscopy there in Panama, or for our project, for our flowering project. Here you see an example. We have some protocols. We went to the field. We clipped or trimmed cut down some samples, and then in the lab we get the spectra from the flowers. And what happened is also like we found two different species. One of them, the Cespedecia spatulata that you see here to the left, that we are able to see from the airborne imagery that we collected with the iris spectrometer as well. So this is exciting because we are able to identify the canopy, from the top of canopy view, some pixels with this yellowing spectra showing there. The second species, which is the one that we wanna like keep on exploring and monitoring, is the Tabebuia Guayacan. So with the Tabebuia Guayacan, as you see here in the example, is super bright. We can find them like along the canal, but also in Panama City. And not only in Panama, we are able to see these species, well, the Tabebuia genus with different species. We're able to see them from the north of Mexico through Central America and South America, even in Brazil. So to see here as well, this is from iNaturalist. And also something very important to highlight here is that because these are located along mid-latitudes, here the seasonality is different. Here the seasonality depends on the dry and rain dynamics. So we have two different dry and rain seasons, let's say, in these latitudes. So we have two different blooms, or two different peaks of bloom. One around March, April, and the second one around September. As I mentioned before, what we are targeting is the genus, the Tabebuia genus, which has several species with different common names along Latin America. In Mexico, in Central America, it's called Guayacan. In Colombia, it's called Cañaguate. In Venezuela, Araguane, and each and every country has a different common name, which is interesting because somehow also shows the diversity of along our countries. And here is an example of Colombia. This happened a month ago, the beginning of March. The Cañaguate, how it's called in Colombia. So you see here how we can identify on a Sentinel-2 image. We can see this yellowing area, this blooming area. Towards the north, the Caribbean region of Colombia. And right now, like during the last couple of weeks, from the first week of April, what we are seeing is the blooming of the Guayacan amarillo in Panama. So here just some examples from social network, how people get excited and started to get even more professional pictures from these trees there in Panama. But again, this is the purpose of this call. Like, well, it's great to have this information from social network, but we want people to join us and to help us to register these observations through GLOVE. And we can use the form of trees So yesterday I made an example. Obviously, we don't have guayacans here, and this was in front of my house, but just to showcase how it would look like. So what I have in front of my house is a magnolia. So we use the trees form to get the observation of this flowering tree. We get the time, some surface conditions. I guess most of you know about this. And here we go. So we have the tree, which is flowering right now. Basically, it's like registering any other tree, just that is flowering. That's the only difference, that is flowering. And okay, then what we can register and what we will look for is like registering the Tabebuyas, like in Latin America, together with setting. I'm gonna come back here. Here. So registering the genus Tabebuia and registering the species as well with the common name. I think it's the easy way to just to get the information about the observation with the common name. As I mentioned before, each and every country, or even within the country, these trees are known with different names. So just getting the common name will be simple. It would be better, of course, if people get to know the scientific name, but the common name is just fine. Then I will ask for some comments, like in English or Spanish, if we have people from Latin America helping us with these observations, where I will ask people like getting some details about the predominant color, again, like if these are yellow, purple, white, pink, and something related with the percentage somehow of the flowering. These are 100% flowering or 50% flowering, or yeah, just to get to know an idea of the stage of the flowering phase. Yeah, pretty much that's it. I mean, okay. Submitting the observation, and that's it. Well, once we get all the observations together with the other observations that we get from social networks and from iNaturalist, we'll use these observations for our flowering monitoring system. So our flowering monitoring system basically is what I showed you today during the presentation, that has 3 major phases: a flowering alert system, some alerts that we set from social network, that we will set from Globe, that we set from our DAC repository, or some specific areas that we are interested for some specific purposes. Then the second phase will be like science. This happen in a cloud computing environment. So it will be the spectral unmixing that I explained, together with the mapping of these flowering events. And then the third phase of this monitoring system will be like the validation. So globe data will be used for 2 purposes. From one side, to set alerts, and the second purpose will be like for validating our results. With this, I finished the presentation. I'll be happy to take your questions.
I will go ahead and read some of the questions from the chat that people have been writing.
Okay.
I'll read the ones that continue to come in. I will note that Peter did a great job of answering some of the questions,
Okay.
but but I will go through and just and ask again to see if you have any other comments you would like to make. Give me one moment. I am going through a bit of a speaking of flowers, just a mention of flowers has kicked off my allergies. So I am sneezing like crazy right now. Give me one moment. Let me look through.
Okay, let me go through the chat. Are these spectrometers using SIMS or PMT? Can we propose to use the same technology for SIMS or PMT? I'm not quite sure what SIMS or PMT is. So, do you know, Peter? What?
That's fine. I think Peter answered that by
Okay.
saying with the Stella instrument, they can go and make measurements themselves. It's like a handheld spectrometer that
Okay.
was developed by Mike Taylor. So that's something that people can go through and basically go and make measurements using this handheld device.
Oh, I see.
Give
Okay.
me one moment. Anna from Argentina. So you did mention that we need to see how Globe can get a Stella spectrometer. It's important for the number of projects we can do with field measurements and satellite comparisons as well. So this is something that, as mentioned, it's been produced fairly recently, and we're starting to kind of get it out to our community to use to make these types of measurements.
True, right.
A question is, could Gaussian mixture models be used to cluster flowering phenology data and it just moved away, based on location, bloom timing, or satellite indices? Would that help uncover climate-related patterns? And Peter did answer, but I'd like to get your answer as well.
They have project class. Well, the Gaussian mixture models are, let's say these are just classification methods somehow. It's a sort of classification method that we can use for active, passive data sets, or I mean like data collected from active or passive sensors. And as long as we have the observations, we can just run the model. The most important is to have the observations, and for the purpose of phenology, we rely on time series and high frequency to see the phenology and to monitor the phenology. So the example that I show with the airborne campaign in California, we were able to see these patterns because, as I mentioned, we collected weekly imagery from that area. So usually flowering phenology or the flowering phases last, some species last just few days, like 5 days, and that's it. For some others last longer. So the higher time frequency that we have, the better for this purpose of monitoring phenology. Then the Gaussian mixture is just clustering method, basically.
One question I have is, when will our community, or where can they find out when these types of flowers and plants are going to be blooming so they can get their students ready to go out and make these types of measurements? Is there something on the website or somewhere you can point them to say, during this time of the year in this part of the world, this plant will start to bloom. This is when you want to go out and make these types of measurements.
We should be able, I mean, we should do that. Yeah, we should give these indications and help people to get an idea what we will find in a specific place based on what we already know. Obviously, the idea is also like to expand this to other species. I mean, like we can just keep on monitoring some other species around the world, but for this purpose, for the purpose of using Tabebuia and to monitor dynamics or environmental dynamics as we do with the cherry blossoms, yeah, for sure. We can just indicate a couple of areas. Mostly, I guess, from what I've seen in social networks that help a lot. I mean, I have like cluster areas from social networks with a heat map to just see, like in Mexico, Oaxaca, there's always like something happening around the blooming season there. Or in Panama City, or in the area that I showed you in Colombia, or there are a few places where there's always people like reporting on this. But the idea is also like to give the freedom to more people to just to let us know, because perhaps also what we're seeing from this social network could be biased, no, on access to internet or to, I don't know. So just thinking of different ways of how to reach some other areas where we are not able to reach.
Okay. Another comment has come up saying trumpet flowers like the Matsaguta, I probably butchered that pronunciation, and Palo Blanco only bloom once a year in Guatemala. And they also mentioned that the Jacarandas in Mexico City, apparently they bloom there once a year as well. Just a comment, really.
Yeah.
And a lot of thank yous for your presentations as well.
Oh, thank you so much for your attention, for joining me today. I mean,
Do you have any other recommendation or advice when people are going out making their measurements? Because one thing we always say is make sure when you're making your measurements to be safe. That's the main thing. But also, I think when we talked when you came to visit JPL, I mentioned that when we had a really big bloom, I noticed a lot of people went out, and it was great to see so many people taking pictures of a lot of these flowers. But I did notice a lot of people weren't being very environmental as they walked over flowers and they trampled on things just to get their picture. Do you have any advice for people who are going to go out to the field and make these observations for you to make sure that they do that not only in a safe way, but to have like a very low environmental impact on the area?
yeah, definitely. So I know we get excited about seeing these flowering events happening, and we want to take the best selfie we can and share this with everyone. But yeah, let's keep in mind that these are fragile environments as well that we want to conserve and preserve. So just make sure don't step on the flowers if these are like grasslands or shrubs, and don't try not to like grab them from the trees. This is an anecdote from last weekend, 2 weekends ago, here in the cherry blossoms in DC. I went to the cherry blossoms area and I saw people shaking the trees to get a selfie, like with a kind of like a rain of flowers. I was like, no, please don't do that. So just be careful and be aware of that what we want is to preserve them and keep on like admiring them for way longer. So yeah.
So that way, don't shake trees. We have another question. Somebody you might know, Dr. Erica Podest, who accompanied you on your trip to Panama doing her research. She asked a couple questions, and one of them is curious why yellow is the predominant flower coloring. What does it mean for pollinators? That's one question.
Yeah.
Uh, and a second question is how far back are you going in time to note the timing of the blooms with satellite imagery?
Yeah. Okay. So for the first question, I think, as I mentioned, the main purpose of flowering is pollination, not reproduction. So they have been evolved in such a way that they want to show the colors or display the chemicals that most pollinators are able to see. And it happens that most pollinators are able to see UV and have a channel located for a spectral band, let's say, or a channel located in the yellow spectral range. So I guess the contrast, this is just guessing. I have to learn more about it, but guessing that the contrast between UV and the yellow spectral band or the spectral channel will play a role in the pollinator's vision. So I guess it comes from that. But yeah, this is just a guessing, something that I have to keep on learning from plant scientists and botanists. So I make my best
And,
time to answer this question very well.
yeah, and she has
Uh,
one more. She asks, could SAR, synthetic aperture radar, help with your research?
I think, yeah, especially for those areas where we have cloudy conditions most time, like in Panama, precisely. So I was trying to look for some nice examples for today's presentation, and I found a couple, but somehow cloudy, and some tried to get confused between yellow and some other let's say shadows from the clouds. So yeah, like SAR could help a lot, especially let's say tracking some of the canopies, because when maybe I mean, I'm not sure how it will look like flowering on a SAR image, and I have to learn about SAR technology. But for sure, monitoring the species or this sort of species as well, like not necessarily when they are flowering. So yeah, I mean, looking forward for seeing how to use alternatives for monitoring flowering phenology.
Oh, and Brian shared some information about SAR, synthetic aperture radar, for those who aren't familiar with what that is, in the chat box. So I just once again like to say thank you for having. Oh, go ahead. Looks like you have a thought.
I was thinking about something like, yeah, something that I missed to mention is like flowering happens following a recipe, basically. So the right temperature and the right amount of water. So sometimes, like in these areas, like California, these areas, we're able to see large displays of blooms when these species get the amount of rain necessarily to bloom, and also the right temperature. So thinking of the connection with SAR, I mean, if we can somehow get an idea of humidity or wet conditions on soil, we can also start to learn what are those conditions that help flowering to happen somehow.
That is great to know, because as Erica pointed out in the chat box, we have a mission called NISAR that's going to be launching in the near future. Hopefully the data from NISAR can help with the research, as well as other researchers as well. So with that, I'd like to say, I know we're short on time. I'd like to say once again, thank you for being a part of our special webinar. This is great because we don't get very many speakers to talk about your expertise. So once again, just like to say thank you for joining us. And I'm going to go and turn it over to Brian, because I think he has some closing remarks. And once again, if you want to monitor the chat, there may be some questions that we missed that maybe you can help answer.
All right, thank you. And one of the things that I heard just, which was amazing from Dr. Angel, was that she mentioned that she's still learning about this thing. And that's awesome because, as you know, scientists, when they're doing their research, they are experts in that research, but they are always learning with every step. So I want, and that's a good message for our students who are part of the campaign, the educators who are part of the campaign, who are working with the students with their student research projects every year for GLOBE and the International Virtual Science Symposium every year, that the students, they're taking the measurements and they're always learning about what their measurements mean and, what they mean in the real world, things like that. So I just thought that was awesome to hear from you. Thank you for that, you're always learning. We're always learning all the time, but it was great to hear that because, you're an expert in this field, but you're always learning. So excited to see that. So I'm going to finish off with a couple slides here in just a couple minutes. But here's just a reminder of the Tabebuia tree, the genus that you're looking for, and this is what was mentioned on some of Dr. Angel's slides there. And there's basically phase 1, phase 2. Phase 1 is now through May, so next month, and then phase 2. Primarily September, but we're just putting in here August through October because you may have early blooms, late blooms, things like that. So, and it's just this map here is of the GLOBE Latin American Caribbean region. which I believe, Dr. Angel, you may be doing a presentation for the Globe Latin American, Latin America and Caribbean region around May 15th, and I know that's in some planning stages right now. So, folks in that area, stay tuned for that information about that. I know you're working with Mariana Savino with that coordination office for the Globe LAC region. So, you know, everybody stay tuned to that. I'm sure in that region, you'll see some information about that in the near future. And then also, I just kind of grabbed a couple pictures here just to showcase this. Some of the ones that they're focusing on, you know, they do look a lot very similar, and that's where it can be a little tricky. So with that said, and I know that you showcase this a bit in your presentation, that when you're taking like a tree height observation, you can go in and pop in Tabebuia tree, and that will pop up. The different ones will pop up. But then, you know, if you're unsure of that, and you have seen that blog that I wrote back in April that we highlighted at last month's webinar, that no, sorry, it came out of March or beginning of April. I don't even remember anymore. So many blogs going on. But this blog was about Globe Observer trees. Just got even better, where we ask you that as an optional observation to enter the genus and species. And if you're unsure of that, you know, we ask you to potentially use another a tool, you know, an app like iNaturalist or Seek by iNaturalist or TreeSnap to identify those trees and make sure that you're identifying the, you know, that correct, you know, Tabebuia tree genus when you're taking your observations. So excited to see that. And I'm going to finish off with this. This is at our regularly scheduled Trees Around the Globe Student Research Campaign webinar will be on the 29th of April, 2025 at 1 p.m. Eastern Time, which is 1700 UTC, 1900 Central European Time, Standard Summer Time. And it's going to be the State of Trees, April 2025. Trees and their impact on carbon storage and surface temperature, a comparative student research study of trees in Argentina and the United States. And that is with Ana Prieto. She is on here today. So thank you. I'll be talking to her in the near future about this as well. And then we're also going to have, and there's some folks on who some had to leave and some maybe still on from Globe India. They will be doing a special presentation from the SNJB's College of Engineering India and some of the work they've been doing with the Globe Clouds and how it fits into the whole Globe Observer and Trees Around the Globe Student Research Campaign. So stay tuned for that. So I want to thank everyone for being here today, and this recording will be available soon once Zoom gets it to me. And then I will then send out an email with that information about it when you can access it, and it's going to tie in. It's going to be on the Trees Around the Globe Student Research Campaign webinar page that you can access that. And once again, thank you, Dr. Angel, for your amazing presentation and getting folks to get interested in seeing this color from the ground, from airborne, from space, and you know, and the request for Globe observers and students, educators, anyone using Globe Observer, Land Cover Trees tool to find these trees, you know, as part of your research. So thank you all so much for being here. And until next time, everyone, take care.